Thanks, Andrew. Okay, it's my great pleasure to introduce today's seminar speaker, Nicole Millette, here from VIMS. So Nicole is from originally from Minnesota. You were talking about before we got here. She went to undergraduate at University of Rhode Island, where she worked with folks like you worked in Susanna Mendendoyer's lab.
I worked with Liz Harvey.
And with Liz Harvey, right? So she's been doing protist plankton work for pretty much her entire career. Following URI, she came to Horn Point, where she got her PhD. She entered here with Diane Stecker. And for the graduate students in the room, you can talk to her about how she made her way here. She pretty much self-funded that entire project. She started as a master's student with Diane, switched over to become a PhD student with me. She finished in 2016. Yep. From there, she took a postdoc in Miami with a joint postdoc with NOAA and Mississippi State University. From there, she moved to Hui and worked on protists with Becky Gass, primarily in the polar regions. No?
My work was.
Your work, what Becky Gass does most of her work in the polar regions. You were working in Wacoit. Yes. Okay. I'm getting it right. Now she ends as an assistant professor. She's got a lot of stuff. She's at VIMS as an assistant professor, and today she's going to talk to us about her work on mixotrophy. Thanks for coming.
All right. Here we go. All right. Great. Hello. Yes. Thank you, everyone, for coming. Yeah. So last time I was in this room giving a seminar was my defense. So if anyone wants to go into the side room over here and ask me a couple questions for a couple of hours, that's great. It's just like old times. But yeah, so I started my position at VIMS in February 2020, so pretty much the start of the pandemic, which gave me a lot of time to think about things and then get people together to think about things. I've been thinking a lot about mixotrophy, and I'll be talking a lot about the stuff that we've been discussing in a working group I've been a part of, and then also sharing some of the research that I've done, and then wrap up with kind of going forward with So for people who are unfamiliar, a mixotroph would be a type of plankton that's using phototrophy and phagotrophy to obtain energy and grow. So they're just a type of plankton, photosynthesizing and eating. And that used to be, the idea was that you still have your main groups of phytoplankton and zooplankton, and they are sticking to strict photosynthesis, strict ingestion. And then you had these, quirky little organisms that were mixotrophs, but there weren't that many of them. And thanks to a lot of scientists, especially Diane here at Horn Point, it became clear that it wasn't as abnormal for an organism to be a mixotroph, and a lot more of them were actually capable of utilizing both nutrient modes to some degree. And so what this figure here depicts is this idea that all of these different types of protists fall on a spectrum, usually between photosynthesis over here and phagotrophy over here. So stuff will still be your strict photosynthesizers or your strict engrazers, but a lot of stuff will express some degree of both of them, even if it's just a little bit. And there's definitely stuff that falls in the middle here where they're really showing capabilities of using both modes pretty easily. But what this also means is that thinking about organisms as zooplankton versus phytoplankton maybe isn't necessarily how we should be viewing the plankton food web anymore, and we should make space for this third group as a mixotroph. And so that research needs to make that transition, as well as how do we actually do research that targets mixotrophs in the environment and in the laboratory, so that we stop, because it's common, it's a reflex. Even I do it, talking about organisms, and I'll say phytoplankton, but I'm talking usually about organisms that are in fact mixotrophs. So it's a major change that has to happen, but there's good reason for it. And also, when I talk about mixotrophs, it's not just one type of mixotroph. Part of being a plankton group means that there's multiple different functional groups and ways that you can categorize a mixotroph within that umbrella of a mixotroph. And one of the most common ways, or most accepted ways these days, is four different categorizations that are actually broken down into two different groups. So this first group here is what's called a constitutive mixotroph. This might be what a lot of people think of when they hear mixotroph. So this is an organism that has their own chloroplast. And when photosynthesis is not ideal, or they're stressed out due to a lack of light or nutrients, they have the ability to start ingesting other organisms. And then this group here, these three organisms, all belong to a category known as non-constitutive mixotrophs. They don't have that innate ability to have their own chloroplasts. In some way, they have to acquire chloroplasts from other organisms. And two of them here, you have your generalist and your plastidic specialist. A generalist here can steal chloroplasts from a wide range of prey. It can't hold on to that chloroplast for very long. It can use it for maybe a couple hours, and then it'll start to break down, and they will either digest it or kick it out of the cell. And these plastidic specialists, this is going to be something like a really common example would be Mesodynium or Dinophysis. They steal the chloroplast from a very specific prey, and then they hold on to it actually for a while, and they have some own capabilities to maintain that chloroplast inside their cell. And the final group over here is endosymbiotic specialists. So this is actually going to be much larger organisms, something like an acantharian or radiolarian that has endosymbiotic relationships with phytoplankton, with phototrophs inside their cell. And it's more of a symbiotic relationship than stealing chloroplasts. And it can get really fun if they're endosymbiotic. With their symbiont is also a mixotroph. That's a whole other level. And the reason it's really important to focus on studying these mixotrophs as a group is how it's likely that they can impact the flow of carbon in the food web. So this is from Ward and Follows. This is a famous paper from a couple years ago at this point. But what they did was set up hypothetical plankton models on the global scale and looked at one version of this model was things that are strict phototrophs, strict heterotrophs, and the other version is everything's a mixotroph, and it's going to decide whether it's a mixotroph based on the size of the organisms. But when mixotrophs were in the model, and the model were mixotrophs, what was happening was there was increasing trophic transfer, so more carbon was moving up to higher trophic levels. And the reason for that is because now primary production can be happening at these higher trophic levels. So all carbon's being lost, a lot of carbon's being lost to respiration as it moves up, but some of it now can be replaced if some of those higher trophic levels are in fact photosynthetic. And because of that, the model suggested that then when there was more mixotrophs or all mixotrophs in the food web, in this case, the size of organisms would then be larger, more biomass would be stored at these larger organisms, and that would also impact increase carbon sequestration and transport down to the bottom because we got larger organisms. So if mixotrophs are a big part of the food web, then we might be misunderstanding what the food web actually is and how carbon is moving through the food web. So that brings me to my talk here, the meat of my talk here. What is, like, how have we thought about mixotrophs in the past and how that informed how we studied mixotrophs? Where are we right now and how we're expanding, how we're studying and thinking about mixotrophs, and then looking towards the future, what are some other studies that can be done that are really, really large scale and more inclusive of just plankton ecology in general. And so that mixotrophy research isn't something that is a group of specialists is working on. It's just a normal part of plankton ecology. So starting with, just this is very brief section here. How a lot of stuff was done for many years was laboratory experiments, and it was looking at these constitutive mixotrophs for the most part, what's causing them to increase their ingestion, what factor is stressing them out. So this example here has 4 different Antarctic species, and some of them, their increases in grazing are responding to to low light availability, or to a combination of low light availability and low nutrient availability. And this type of research was done on it was very common to just take organisms and see, is this a mixotroph? Under what conditions does it switch to grazing? And even I did a little bit of research here on the winter dinoflagellate here in Chesapeake Bay, showing that their ingestion rates increase at low light levels. So these types of these single species studies, they were really, really helpful, and they still are helpful in identifying mixotrophs and the possible niche that they can exploit when that they could dominate in when conditions for other phytoplankton are less favorable. And this type of research is also really, really provided that base that was needed to help understand like the different groups of mixotrophs and how different mixotrophs function just as a group, and has really helped with that categorization. So to be clear, I am not saying that this work is like in the past and no longer needs to be done. It's just we need to expand off of this and be doing more. And one of the things that this type of research does is it misses the mixotrophic community. So, I mean, it's just, we're talking single species, but it was part of that idea that these were just unique types of species that were not common occurrences. But if they are a common occurrence, then we need to be looking at, well, who's this mixotroph community as a whole? And there was research that was using fluorescently labeled particles, either heat-killed bacteria or plastic microspheres, and feeding them to a natural population, and then looking underneath the microscope at who was taking up these beads and estimating ingestion rates, and then also estimating the abundance of the mixotroph community in comparison to the heterotroph community and the phototrophic community, the strict phototrophic community. And there was been a fair number of papers on this spread over a couple decades, so it wasn't uncommon. In this example here that I pulled out, this is looking at comparing the consumption of bacteria by phytoflagellates, in this case mixotrophs, compared to heterotrophic flagellates in the Mediterranean. And they've got a size components. So this is focused here on nanoflagellates and smaller sized nanoflagellates, and same thing here. And what's really seen here, it's the Mediterranean, it's a much more stable environment. So you're actually not seeing a lot of variability throughout the year in which group is doing more grazing. And there's just, when it comes down to it, the grazing attributed is about 50% towards phototrophs and heterotrophs. But as I said, there's been a lot of studies that have used this exact approach, which means that this data is comparable between studies. And so it was a recent synthesis by Kyle Edwards at the University of Hawaii that combined all of these studies and tried to synthesize some information out of it. And I'm not going to spend a lot of time talking about this very, very interesting paper, mind you, but he found some gradients based on from latitudinal gradient from the pole to the equator, as well as an open ocean to coastal gradient in terms of the abundance of the autotrophs, the mixotrophs, and the heterotrophs. So, some very interesting data. However, these studies too, again, have gotten us very far and have really, really helped us understand just how prominent these mixotrophs can be in the community, but they're still very limiting. So the first part, when I say community here, These studies usually are focused on the nanoflagellates specifically because they're talking about bacterial grazers, and the dominant bacterial grazers are nanoflagellates, and people will usually just filter through 20 microns so that they are automatically just focused on the nanoflagellates. However, there's a lot of other types of mixotrophs, the constitutive mixotrophs that aren't necessarily grazing on bacteria, as well as the non-constitutives that are completely different from what these studies are getting at. So it's still very focused. And so, but the main bonus of these studies is that they can be compared between studies. So it allows for some larger scale understanding across environments or conditions that just because it's what we've been doing, continuing to do it in some capacity has benefits. And it also comes with, unfortunately, other limitations and drawbacks. The main thing is, as is demonstrated here, this is the ingestion of these different beads by a mixotroph compared to the ingestion of live bacteria. And it's heat-killed bacteria or it's these microspheres, and there does seem to be some bias against them. And something I've experienced myself, it's really hard to get above this like 10% of the community measuring them, identifying them grazing. So it's probably also just an underestimation of the community that we're looking at. So that's in the past, but we're at a very, very exciting time in mixotrophy research. There's definitely an expansion of the people working on it and the questions that are being asked. So I'm gonna cover highlight a couple of that, but then I'm gonna get into the recent stuff that I've been doing finally. So some stuff that I think has just been interesting is people are really getting into using stable isotopes to measure how much carbon mixotrophs are getting from photosynthesis versus ingestion. And so actually the incorporation of nutrients from those different food sources into the cell and seeing what do they like, what do they actually prefer. Looking at the idea of, are mixotrophs a good source of food? Is there a benefit for mixotrophs to zooplankton compared to autotrophs or heterotrophs? Or not, because again, these are now a separate category compared to phytoplankton and zooplankton. And there's been studies to look at, do different copepods like to eat phytoplankton or zooplankton, and what's the impact on their egg production or their habitat, their growth rate efficiency. So now adding in that third category of mixotrophs, how does that impact things? And then also there's been a fair amount of work that's looking at global distribution of mixotrophs and trying to understand just where are mixotrophs important? What environments do they like? Again, this is the kind of thing we've got better understanding, at least for phytoplankton and zooplankton, on these large scale patterns, but we're behind on mixotrophs. And this study here was using a global data set of just microscopy samples that had been collected, and going into these microscopy samples and just saying, oh, well, previous research has shown that's a mixotroph, that's a mixotroph, that's a mixotroph, that's a mixotroph. And then looking at, in this case, more of a presence absence sort of thing. And this is a Tara Oceans data set. So this is sequences, and this is looking at going back to the sequences and saying, well, that sequence is associated with a mixotroph, that's a mixotroph, that's a mixotroph. And then again, a presence absence of these sequences. Yeah, and so that's how this taxonomic identification is. It's just going in, you got a list of taxa, and you're just reclassifying based on previous research who's a mixotroph. And because there's all of this available data out there from microscopy samples, or now more so IFCB data or FlowCam data, you have the ability to at least estimate abundances and presence of mixotrophs just because we have a better handle on who is a mixotroph. And so this type of data, I really am excited about the idea of being able to just reanalyze a lot of historical data. And if you have data that's associated with environmental data, then you get to do a lot more analysis, and that's very much more exciting. However, there are drawbacks to this approach. And so a lot of microscopy data tends to be biased towards larger phytoplankton. So in this case, we might be missing some of the smaller mixotrophs, but we also, and also depends upon the sampling method that's used, whether you're biasing yourself towards larger or smaller phytoplankton. And just because research at one point showed an organism's a mixotroph doesn't mean necessarily in a sample you're looking at that was collected, some random time point, it's acting as a mixotroph, or it's even has the potential to be a mixotroph in that sample. So that could lead to a possible overestimation of mixotrophs. And so this is what I've been interested in, is can we find a better way to constrain what is a mixotroph in a system? I really identify who mixotrophs are in a certain area and who's acting as an active mixotroph in the natural environment. And so that is, this is something I started working on during my my postdoc up at WHOI. This is something that Becky Guest helped to develop, and I've got really excited about the potential for it. And it's the idea of taking bromodioxyuridine, it's a nucleotide for analog for thymidine, so BRDU, it's shortened to, and you label the bacteria's DNA with this BRDU. You take this bacteria and you feed it to a natural sample of protists, and then some of these protists will ingest this bacteria, and they will incorporate that BRDU into their own DNA. And so you'll have treatments where you have bacteria that have the BRDU, and you have the treatments where bacteria does not have the BRDU. And at the end, with the non-BRDU data down here, you would run your same standard extraction of DNA, PCR, sequencing, and get your amplicon sequence variances from this data set. However, with this stuff that was fed BRDU labeled bacteria, what you have to do is this aminoprecipitation process, where what you are doing is taking any of the DNA that has this BRDU incorporated with it and separating it out from any DNA sequences that doesn't have this BRDU associated with it. And this is like a 3-day process. It takes a very long time. There's a lot of times where it just has to sit overnight, and you're changing the temperature on it and all of this stuff. So this part's very time consuming. But then when you're done, you have this subset of DNA that you then would run your amplicon sequencing on, your PCR amplification, then you get your sequencing data, your ASVs. And I won't bore you with the great details of how specifically you determine something is a mixotroph with these two different ASV data sets. But needless to say, you can end up with a very long list of what is the sequences that were identified as being associated with a mixotroph, who was ingesting this bacteria and who has chlorophyll in them. And alongside this, if you are collecting your own phytoplankton microscopy samples to analyze, or if you were to have a historical data set associated with the region where you were running these BRDU samples, then you can start matching up things as we would do for just for what people have been doing before, which is what has been known to be a mixotroph, but this is now much more specific to the location. And so this is examples here of data that I collected up in Chesapeake Bay of these were my own microscopy samples, and then a subset of who was a mixotroph. And so, a couple of things. So some of the results then for the Chesapeake Bay research. So this is Wacoit Bay here. It's actually quite small. People have not been to it, and it has a very weird, messy flow system to it. But I was sampling at one of the freshwater flows into Wacoit Bay up here, Childs River, and then one of these saltwater exchanges down here at Minot, and went out for just over a year at these two stations, sampled 1 to 2 times a month. And then for 6 of those dates, I set up BRDU experiments. So that would be 6 at each of these stations. So for every date, I had microscopy samples of what phytoplankton were present. And then for this summer data here, I had data of what mixotrophs were present at each specific station within the system. And over here, these results, this is looking at just the proportion of mixotrophs from the sample dates in which we collected the microscopy samples and set up the BRDU experiments at the same time. So for these specific data points right here, this is only matching up who was a mixotroph that was actively grazing at the time the sample was collected. So that varied a lot from week to week. And you can see here, all it is that between these two stations and between weeks, the proportion of what mixotrophs were actively grazing was highly variable. Down here, this is a lot more messy of a data set, but this is estimating the percentage or the proportion of potential mixotrophs. So this is looking at all of the data, but it was just who was identified at any point as a mixotroph at each of these individual stations. So was definitely allowed for more things to be identified as a mixotroph. And there's a lot of data here, so you can't possibly understand, which is why we did some statistics, and that's what I'm going to actually show you now. So don't focus too much on that graph. But what we did was general linearized models to look at the environmental conditions that were associated with the different major taxa groups, and then the abundance of mixotrophs and the proportion of mixotrophs. And this is Childs River Station, so this is the one closer to the freshwater source. And what you can see is the environmental conditions associated with dinoflagellates was very similar to the conditions that were associated to the abundance of mixotrophs here. So there was a lot more mixotrophs when you had a lot more turbidity. This is NTU. This is when there was higher stratification in the water. Actually, sorry. Oh, I was confused myself. This is a stratification index. This is actually when there was lower stratification in the water. The index was higher, which means the stratification was lower. But there's also less nutrients, specifically the nitrogen, but there's higher phosphate. You do that with across the board. So suggesting that at this Childs River Station, you're getting more active mixotrophs when there's potentially nutrient limitation, but also when you're getting a lot of more dinoflagellates in the water, they have a higher abundance. And then down here at this Minot Station, this is the one with more saltwater exchange. The conditions associated with the abundance of mixotrophs was very similar to cryptophytes. Very, very similar indeed. And this one here, you got more mixotrophs when there was, when it was more in the summertime, so higher temperatures, when there was lower nutrients again, but also this is the one here where you had higher stratification at this station. So when there was more stability in the water column, there was more mixotrophs. I'm not showing the data for it, but one thing is that the proportion of cryptophytes between the stations was the same, but the difference in terms of the community composition came with a switch between dinoflagellates and diatoms. So up here, this freshwater station had a higher proportion of dinoflagellates, and up here, this is where the dinoflagellates were controlled or were associated with the mixotrophic abundance. And down here, there was a lot less dinoflagellates, more diatoms, and probably why the cryptophytes were associated with it. But I mean, I did the best I could do for, a postdoc, a short postdoc. And I still only ran 6 BRDU experiments at each station. I didn't cover all of the dates, so can do better. And luck, so, you know, we'll see if I did better. But what we can see here from Micronutrient Bay, from just better identifying these mixotrophs at the specific station, is that we can see that the difference in the, I should say abundance, sorry, the abundance between the stations, the difference at Child's River and the variability that was very associated with a specific taxa group. Meanwhile, it was associated with a different taxa group at the other station. And so to some degree, the type of species that are present in the community could be controlling the mixotrophic activity, and something that hasn't been necessarily considered. But I wanted more BRDU data. That's a great thing about having students, is you can make them do stuff. So you can suggest that they do some work, and then they're very eager to do that work, even if maybe it was a lot of work. But I had Marcella DaCosta is a master's student of mine who will be defending this spring, if I have anything to say about it. And so she went out and collected data twice a month for a year at 2 stations within the York River here. So this station, Gloucester Point, is right by Bims, just up here, right by Bims. And then this is West Point Station up here, and it's a very low salinity station. So this is actually the Mattaponi. You've got a lot of fresh water flowing in here. This is like 2 salinity was typical here. Well, this is about 20 salinity. So she did pretty much the same thing I was doing at Micronutrient Bay, but now for every single data point, we know who the active mixotrophs were at both stations throughout the year. And so this data here is just looking at this ASV data, who the BRDU experiments identified as a mixotroph. And up here at West Point, so this is just the number of ASVs associated with these different major taxa groups of phytoplankton. And at West Point, what stands out is, first of all, at the beginning of the year, a lot of these ASVs were associated with dinoflagellates. Not unsurprising. Dinoflagellates is the most famous group associated with mixotrophs. Almost all of them seem to be mixotrophs to some degree. However, as you moved later in the year, there was a lot less dinoflagellate ASVs, and there was a lot more cryptophyte ASVs that were showing up. And another group that was really important at this low salinity region was also these chrysophytes. This bright purple group. They were showing up a lot. Down here at Gloucester Point, pretty much the dominant group throughout the whole year was dinoflagellates. There was dinoflagellates, at least grazing. And the other thing is you can see this variability in the number of ASVs that were identified as mixotrophs. So this is starting early in the spring. You get a lot of ASVs that are coming up as mixotroph. And you have to get this dip in the summertime with a little bit of a spike at both stations in late fall here, and then a little bit of a decrease coming into winter. So there's also some variability in the number, potentially number of species that are actively grazing. And this figure here just further emphasizes the difference in the different importance of different groups at both stations. So again, Gloucester Point here, over 50% of the ASVs that were identified at Gloucester Point were associated with dinoflagellates. That was the main group. And dinoflagellates were still the most prominent group within the ASVs at West Point, but this is the cryptophytes and the cryptophytes, and both of them were much more important compared to Gloucester Point. So this is here looking at what's the actual abundance of these active mixotrophs throughout the year, as well as their proportion. And it doesn't necessarily follow the pattern of when there was the most number of ASVs. But at least here at this is active mixotrophs, and the orange is the West Point, and the green is the Gloucester Point station. And at West Point, you can see there's a really high abundance of mixotrophs, specifically in the fall when there was the cryptophytes were the ones grazing. And then at Gloucester Point, it was a lot more sporadic in terms of the abundance. There wasn't really any time period specifically that stood out. And then same thing here with the proportion. The proportion of mixotrophs and the abundance of active mixotrophs really matched up well at both stations. And you can see that in this fall time, it's also when you get a higher proportion of mixotrophs in the community. And there's definitely times when the mixotrophs have a very low proportion, but when they can account for a high proportion of at least the community that contains chloroplasts, it can be, in this case, up to 40% is what we estimated. So that's not nothing out there. And this just gives you a better idea real quickly of who in our microscopy samples were these mixotroph species, in case anyone here is interested in the species of phytoplankton. And so here, I'll start over here at Gloucester Point. So this is just the different mixotrophic taxa in these microscopy samples. There was a lot of gymnodinium was showing up, Heterocapsa rotundata, and a little bit of gyrodinium. And there was some of this telealex, which is a cryptophyte, but it wasn't grazing all of that much at Gloucester Point. But here at West Point, you still get a lot of this gymnodinium. That seems to be pretty universal, at least across the York River. And of course, this telealex, this cryptophyte, is showing up a lot later in the year. So it's this telealex who is grazing a lot. And the thing I want to end with here is to point out that cryptophytes weren't grazing at Gloucester Point, but that doesn't mean there weren't a lot of cryptophytes at Gloucester Point. So here, this is the proportion of the different major taxa groups identified through microscopy. And the yellow here again is these cryptophytes. And just throughout the whole year, with the exception of a phytoplankton bloom that happened here at Gloucester Point, it's pretty much can be dominated by these cryptophytes, but they're not grazing at the station. The grazers are associated with these dinoflagellates down here. Meanwhile, there's definitely cryptophytes at West Point, but they're at times it can be a lot less abundant compared to the Gloucester Point stations, and they're not even necessarily dominating the community in the winter time or the fall time, but they're grazing. They're just constantly grazing. Well, over here, you definitely get a lot of dinoflagellates, and they are in fact grazing. So just because something's present and is known to be a mixotroph doesn't mean it necessarily is a mixotroph. So just to briefly summarize these very preliminary findings at these two different stations is up at West Point here is dinoflagellates are active in the spring and the summer, but it's the cryptophytes that are much more active in the fall. But overall, the mixotrophs were much more important at this station in the fall. Down here at Gloucester Point, the dinoflagellates were active year round. The cryptophytes were present, but not necessarily active. And the importance of mixotrophs can be very high, but it also was sporadic. One thing I do have to mention though, is that because we're sampling at fixed stations off of docks, we can't always access, like a lot of the water. And so one thing we do know for sure, this Gloucester Point station is during these dates here, we missed a large dinoflagellate bloom that was happening in the York River. We could see it from the dock, but it wasn't where we were putting down the Niskin bottle, so they didn't show up in high abundances in our samples. So there's pros and cons to this sampling approach. This is just now overall talking about the present research focus on mixotrophs, not necessarily just my research, but we're really trying to expand the study of mixotrophs over a larger scale and getting at more than just ingestion rates on mixotrophs and measuring what's causing environmental variability when they increase their ingestion, and really trying to also use new methods to address questions and collect data in a new way so that we're able to sample and understand different parts of mixotrophs. And so then very quickly, just going to maybe, I guess, very selfish here. And when I talk about the future of mixotrophic research, talk about the 5 proposals I submitted this last year about mixotrophy and what I've been thinking about. But first of all, so I'm part of this OCB working group that started January 2021, and we've been having these virtual meetings and some in-person meetings. One in-person meeting, but talking about this idea of like future research priorities and then different methods to be using. So there's a lot of names here. We got Pat here, of course. Where is Emily Brownlee's come through here. And so just a lot of really exciting conversations. Oh, and Matt, of course, can't forget about him. So we actually have laid out what we feel are 5 research priorities, very broad research priorities on mixotrophs. And we put together into a paper that was rudely rejected. And so now we will resubmit that paper and try again. But the gist of these research priorities is evolution of mixotrophs. So focusing on just all of the different times along the evolutionary tree that eukaryotes acquired chloroplasts through, stealing them from bacteria and became phytoplankton. And along those pathways, when did things go all the way to just stopping grazing and only having these chloroplasts versus when do things, keep the ability to ingest and steal chloroplasts or have their own chloroplasts, which then kind of feeds into this idea of trade-offs. So a mixotroph having the ability to photosynthesize and ingest inside the same cell is associated with trade-offs. So what are those trade-offs? And for better understanding that, and by understanding these trade-offs and the things that are driving these trade-offs, we can potentially better identify locations in the ocean and by a geographical distribution of where we would expect to find a lot of mixotrophs or heterotrophic activity. And so then we can go out and sample and do research and say, are there in fact a lot of mixotrophs where we'd expect them to be? And once we identify places where there's a lot of active mixotrophs and they're a big part of the food web, how are they impacting the biogeochemical cycling within that system, or the trophic transfer? Are they actually improving the efficiency of trophic transfer and increasing carbon sequestration as the Worden follows model suggested, or not? I don't know. But of course, to do a lot of this research, we're going to have to do a lot better at identifying mixotrophs in the environment. And so, finding ways to come up with new methods or adapt methods that people are currently using to mixotrophs is a big focus. And so one couple of the ideas I've been thinking about is I've been interested at least in this idea of jointly measuring the amount of carbon coming from both photosynthesis and ingestion for overfeed. I've termed it a trophic ratio, the ratio of carbon coming from phototrophy to phagotrophy. And in this case, using stable isotopes of inorganic CO2 and CO2 labeled, stable isotope, the C13 labeled prey. But the hypothesis actually being tested for this research would be that these smaller size cells are a lot more phototrophic because they have a really high surface area to volume ratio, so they should be really good at taking up nutrients. And then these larger cells should much more favor phagotrophy because they just be bad at photosynthesis. But then in this middle area is where you're going to get this trophic ratio that falls in this middle, a lot more like your mixotrophic organisms. But the other important aspect is how much variability along this trophic scale, this trophic ratio scale, these different sized organisms can move. So really small organisms, really large organisms, we hypothesize should be very tightly constrained to their preferred method. No matter how much you stress them out, even if they can use an alternate nutrient mode, probably not using it too much. Well, these organisms in some kind of median size, whatever that ends up being, is likely to be able to fluctuate wildly between these two nutrient modes is the idea. And we were told this would be recommended for funding, so it should start next year. That would be exciting. Another project here. I really like this one because it's a lot of measuring the change of ingestion rates in the lab on different organisms, which I said was a very, like old school way of doing things, but we're adding on a lot of new methods to understand these specific organisms that have been really well studied in the Antarctic. So the specific thing we're looking at is, do these organisms respond to micronutrient limitation in the forms of iron and manganese, which is an issue down in the Southern Ocean. But it's not just looking at changes in ingestion. We're looking at how does their transcriptomics respond? And then actually, are they filling their intracellular nutrient quotas for these micronutrients when they are ingesting? Is it giving them the nutrients that they actually need? And then hopefully with these transcriptomics, being able to come up with some identifiers of mixotrophic activity that could maybe then be applied to the field. We'll see about that. But luckily that's not a requirement for the project. And this is one we've been told is being considered for funding. So maybe also starting next year. I don't know. We'll see. It's not rejected right now. So that's good. Then another one also in Antarctica is there's been a lot of research. In this case, is really looking at this idea of like mixotrophs as prey quality. I'm very, very interested because I've always been interested in like trophic transfer and like zooplankton feeding on, smaller plankton. How are mixotrophs as a food source? And so this one, the idea is to do it proposed to do it in the West Antarctic Peninsula where their LTR is, but this is research that could be done anywhere. And there's a lot of different things I want to look at, like looking at how the CNP ratio and fatty acid composition of mixotrophs change when you stress them out. How does feeding, stressed out mixotrophs compared to stressed out phytoplankton impact copepod ingestion and egestion? And that doesn't necessarily have to be done in Antarctica. Also in Antarctica is the idea that there's actually been a lot of samples collected along this LTR related to DNA. And people have already done analysis on a lot of these samples. And what they've showed is that there's a shift in the what they say is the phytoplankton community from warm years compared to cold years. But that means these samples are ripe to going back and reanalyzing things to pull out mixotrophs, known mixotrophs from them that exist along the West Antarctic Peninsula. So this would be done using qPCR analysis on these samples that have already been analyzed and actually trying to estimate abundances of a range of mixotrophic organisms and seeing how do these mixotrophic organisms respond to climate change. This one will definitely have to be resubmitted. There's a really funny story about how it fell apart at the end and probably shouldn't have been submitted, but I ended up as a lone collaborator on a project I had no business being the lone collaborator on, but I'll try again. And I decided to put together a career as well. And this one I'm excited about only because this is taking this BRDU method that I spent a lot of time talking about. And what if we did it for all of Chesapeake Bay? What if we went out and we identified who the mixotrophs are throughout all of Chesapeake Bay, created lists at each of these stations of what mixotrophs are present and at what time of the year they are actually grazing, and did a lot of analysis with just the ASVs But as you might recognize, these are all Chesapeake Bay program stations. So then go back and reanalyze all of that phytoplankton data, look at long-term trends of mixotrophs, their proportions, their abundances, at all the environmental factors, and I want to create these beautiful figures. I've got big ideas, but unfortunately, it is a career. It is highly based in Chesapeake Bay, but maybe at some point I'll be able to start to chip away at some type of this research. And this I'll just end on is a figure we put together for our working group, which is our dream cruise of everyone on there doing something to study mixotrophs, just the mixotrophy cruise we want to have one day. And I will stop with that. We have questions? Yeah, Jacob.
BRDU method, it seems like you'd have different mixotrophs grazing to different extents, and they would have different amounts of BRDU. I'm presuming it's some sort of cutoff, and so if they've incorporated at least as much in their mixotrophs.
Yeah, so the way we identify mixotrophs at least is you have the two different data sets of ASVs. So you look at the proportion that a sequence took up in the ASVs that had the BRDU incorporated and compared them to the proportion in the samples that didn't have any BRDUs, so it's the full community, and they should be any mixture should be overrepresented in the experimental set. And I think the cutoff is if they have if they're overrepresented by like 1% in that one, then they are a mixotroph. So even if they're like way overrepresented, then they're still a mixotroph compared to something that's just barely over.
But there's additional information there, but sort of how much stuff they're taking out, right? They're very represented.
Yeah, well, that's potentially, because you also have to run into like, is it, are they just taking up more bacteria, or is it just the sequence itself being over amplified in the data set for some reason? But that could maybe be teased apart. Yeah. Yeah. Okay.
So, the really cool result of Warden Follows in 2016 was this idea that, mixotrophs are really important in pushing carbon up the trophic transfer scale. And as far as I can tell, are we getting any closer to testing that hypothesis at all? Are we still, I'm sorry, you just kind of tried to talk about that, but it remains a really big open question, right?
Yeah.
So what is there? Do we have any methods for kind of trying to get at that?
So one of the things one of the things we've in our working group we talk a lot about how we can combine methods to maybe get at that. So it could be something along the lines of looking at the just for example the BRDU method or something where you're understanding how many mixotrophs are present or what proportion of that community is actually a mixotroph, and then just looking at maybe the size distribution of that population and comparing between different regions. So if more mixotrophs are present, is the community on average larger, is more carbon stored in a larger biomass? Could be one possible test of at least a part of that hypothesis. Carbon sequestration, well, because the other thing with the Warden Follows was with the higher trophic transfer that led to higher sequestration was part of it, but that's something that we haven't touched on too much and haven't had a lot of discussion about it. But there is interesting questions of how can we understand the impacts of mixotrophy and mixotrophs on carbon sequestration as well. That's another question, and I don't have an answer for that one at least. Yeah, Tom.
I'm curious about the method. Does the incorporation of BRDU make the prey more available or less able to survive? Is there a sort of a
is there a downside to them having the BRDU? Maybe the light causes the BRDU to kill the organisms, and so you can't run the experiments for too long. So yeah, actually the BRDU is light causes the BRDU to be toxic. It's actually a downside to it. But so you keep the bacteria once you incorporate them into the BRDU in the dark, so the bacteria don't die. and then you add them to the bottles, and you've got 24 hours is the only amount of time that they have. So it's more of the fact that you might, if you run it for too long, you might lose a lot of the bacteria, but for that short period of time, if we're not aware of there being a negative consequence on the grazers, at least.
Yeah, I was thinking more about the prey, if the
Mhm.
prey were somehow more, if there would be a bias because somehow they were more accessible to be grazed upon by mixotrophs.
Not that I'm aware of. That's not one of the many issues that we're trying to work through with the method at this point. There's plenty of other things to be concerned about, but that's not one that we're worried about at the moment.
I have one other question.
Yes.
This whole area of research reminds me of research that was done a couple of decades ago when the role of bacteria in the oceans was being investigated.
Mm-hmm.
And, you know, the sort of augmentation of the trophic structure by the bacteria sort of feeding into higher trophic levels. And so, you know, I think our food chains are getting really complicated now. And so,
Yes.
you know, have you been able to draw any analogies between the role of heterotrophic bacteria
Mhm.
and within trophic structure, as well as these mixotrophs within the
Yeah,
trophic?
so there's a couple thoughts on there with like how they're like could be impacting or changing the structure of the food web. And the first one is you have these organisms in the food web that are doing like both an entry point for new carbon. And they're also a point in the food web where recycled nutrients can move up, or at least repackaged, you know, nutrients from bacteria and other organisms, secondary production can go up. And the important thing is that the amount that they're doing either of these, you know, using each of these nutrient modes is variable based on environmental conditions, prey availability, and other things where if you have a phytoplankton that's present, it's photosynthesizing, you have a grazer present, it's eating, but then you have this point in the food web where their activity is less, it's more fluid and also potentially in a lot of ways beneficial, because I've talked about being an entry point for photosynthesis allows for more carbon to be coming up. But going back to heterotrophic bacteria, people have used this term like mixotrophs are a shunt. They're a point where it helps more quickly get the bacteria up to higher trophic levels, the carbon from bacteria at least up to higher trophic levels. It's an idea that mixotrophic scientists have thrown around. I don't know how well it's been tested that they really are improving and increasing the transfer of bacteria from smaller organisms to larger grazers, but those would be major changes and impacts. Yes.
Thank you, Nicole. That was a really nice seminar. And you've made some amazing advances in this field. I'm wondering, is there any generalizable, probably not, but understanding of whether previously thought of phytoplankton are ingesting particles or prey for carbon versus nutrients? Is there?
That's something I really want to dig into with stable isotope methods. And that's definitely a question we've been talking about with this biogeochemical cycling impact is from their old, like, what nutrients are they actually taking up and incorporating from both nutrient modes? Are they actually getting carbon, or are they actually getting nitrogen or phosphorus, or are they getting, you know, maybe even like a fatty acid or something from there? And so this idea of like, I want to track, eventually, this is way down the line, you know, like track where the nutrients that a mixotroph is taking up, where from both sources, where it's going in the cell and whether it's available to higher trophic levels. And then also what nutrients are they taking from their prey? But that's something that hasn't really been explored at the moment.
Great. Well, I think we're about at lunchtime.